Okay, let's begin. Again, hello everyone, and welcome to today's webinar. I am Marissa Fraser Morera with Pion, and I will be moderating today's event. So this webinar is titled Predictive Potential and Operational Use of SCISSOR to Streamline Subcutaneous Biologics Drug Development, sorry. And will be presented by Misha Murashov, Associate Principal Scientist in Preclinical Development in the Biopharmaceutics Group at Merck. Hello, Misha, and welcome. We also have Aparna Chakravarti, Senior Scientist, Preclinical Development and Formulation at Merck, and also Connor Gomes, our development scientist at Pion Inc. Welcome all. Dr. Murashov has worked in the biopharmaceutics group at Merck and Company since 2019, and he is currently one of the subject matter experts on the SCISSOR instrument at Merck. He also represents Merck in the Subcutaneous Consortium. Misha graduated with a double major in biochemistry and biology from Adrian College in 2014, and he received his PhD in pharmaceutical sciences from the University of Michigan back in 2019. Dr. Chakravarti is a senior scientist in the sterile and specialty products group at Merck and Company since 2022, and she primarily focuses on the design and development of sterile products for biologics, small molecule, peptides, and protein large molecule-based drugs for injectable routes of administration. Aparna received her bachelor's in biotechnology from SRM University in India in 2015 and went on to complete her PhD in bioengineering from the University of Kansas in 2022. Lastly, we have Dr. Gomes. He earned his bachelor's in chemistry from Emmanuel College before earning his doctorate of chemistry at Northeastern University. He specializes in the synthesis and characterization of biomaterials, and since coming to Pion, Dr. Gomes has led the development of the artificial extracellular matrices found at the heart of Pion's SCISSOR instrument. Again, welcome to all our presenters. And now just quickly, just want to go over a couple things and we'll begin our presentation. We want the webinar to be interactive and encourage you to contribute to its success by asking questions at any time during the presentation. And to ask a question, we ask you to use the Q&A widget, and it should be located on the side of your screen. We also are going to try to answer as many questions as possible, and whatever we don't get to, we will reply via email after the event. If you need technical assistance or if you run into any trouble, send me a chat message, again with the chat widget. And also I wanted to mention that the webinar is being recorded and will be available to you all within 24 hours or so. And now, without further ado, I will hand things over to Connor, who will be covering the first part of the presentation. Over to you, Connor.
Thank you for the introductions, Marissa, and thank you, obviously, Misha and Aparna for joining us here today. And I'm gonna, again, I'll just introduce myself again. My name's Connor Gomes. I'm an R&D scientist at Pion, specializing in the development of the SCISSOR platform. So today, we're going to be going over a couple different things. Okay, nice. So first, we're just going to briefly review the drug development process and the methodologies used there, so we can discuss where SCISSOR fits into that pipeline. We're then going to review the SCISSOR platform and its capabilities. One moment, there's a delay on the screen on my side. Okay, and the other analytical methodologies that are used here within. And then hopefully for the bulk of this webinar, we will be going over separate case studies and research projects that both Merck and Pion have done with the SCISSOR instrument that's related to the webinar subject today. Now, as many on the call already know, during the drug development pipeline, in vitro and in silico methodologies are implemented during that preclinical stages where you are exploring the API's characteristics or a formulation's characteristics before putting it inside of a human subject. Normally, as we go down this pipeline from drug discovery to drug approval, there is more risk and more cost as we move down this pyramid. And therefore, it's very, not only for scientific progress, but it's also very beneficial for financial efficiency for us to do better at creating in vitro methodologies that are better related to the clinical responses we see thereafter and later in the process. So we have a couple examples of different Pion technology over on the right-hand side that fit into that preclinical window of drug characterization and formulation characterization. But specifically, we're going to be reviewing the SCISSOR instrument. So in the SCISSOR instrument stands for the Subcutaneous Injection Site Simulator. Inside of the SCISSOR instrument, you will have 3 independent experiments that will all have their own methodologies and controls throughout the duration of the assays. We'll focus in on one just for the sake of conversation, but everything we talk about hereafter is multiplied by 3 within the instrument to give you an N of 3 when doing these types of assays. So if we zoom in on one chamber, you'll see that we have an outer chamber surrounding an inner chamber with our cartridge, which we'll get to in a moment. So the cartridge is what will contain the artificial extracellular matrix. This is a simulated ECM environment that's supposed to mimic the biomolecules and reactivities that are present at an injection site within a human at the subcutaneous space. Within this cartridge, it is going to contain that ECM, but also have 2 permeable membranes. These membranes will allow diffusion of material turnover and release of an injectate from this simulated human environment. So on the sides, it will be still the typical clear UV plastic that we can be able to get measurements through, which I'll mention in a moment. And on the front, again, is those permeable membranes that the injectate will leave to indicate uptake into the human body. So in order to introduce your sample into the artificial ECM, there's an injection port with a typical prefilled syringe size injection port. There are, yes, and after that injection, it will diffuse through the cartridge membranes into the larger chamber, which again will signify, which will then be related to the uptake into the human body. The outer chamber is controlled, pH controlled and thermally controlled bicarbonate buffer. That bicarbonate buffer is monitored by a pH probe that will be there during the duration of the assay. There will also be a pH probe monitoring the pH at the injection site. This is used to show the pH changes from the physiological pHs to your injectate pH, and then the return thereafter, signifying the diffusion of that injectate away from the injection site. There's a thermocouple that is typically used to keep the system at 34 degrees Celsius, the temperature of the subcutaneous space, as well as a stir bar at the bottom to ensure that we have a homogeneous system when we are making measurements in that outer chamber. In order to monitor that chamber, we can do one of 2 things. The first is auto sample through a HPLC vial in typical fashions. And the second is through an in situ fiber optic probe with our rainbow apparatus. This rainbow apparatus will have separate UV probes that will all be hooked up to one UV source. And during each of these assays, that is how we will be able to track in situ how much of the injectate is leaving into that outer chamber. But again, if that's not possible because of a UV signal or spectroscopic anomaly, a robotic fraction collector can do the job for you as well, for then offline analysis thereafter. Within the cartridge, although we're monitoring pH, the one other thing we are monitoring within that space will be the opalescence or the turbidity of that simulated ECM environment. There will be 4 different channels signifying that letting you know about the transmission in 4 different parts of that simulated ECM environment. And as your injectate interrupts each of those LEDs, there will be a detector response showing that there's a blockage of light as that bolus moves down and diffuses through the cartridge. These are not only, we also have optical methods of analysis where we will actually be able to monitor and see what's going on in the visible space with cameras that are mounted in front of each reaction chamber. They're examples of what your baseline cartridge would appear as, and then when an injectate is introduced into that space, and then as that injectate either equilibrates within the cartridge or leaves the cartridge, we can see that if the miscibility isn't too much of an issue. So overall, in the SCISSOR instrument, as far as what we get out of it, after introducing a injectate into a simulated human ECM environment, we track the concentration via in situ fiber optic probes of release from that simulated ECM environment, signifying uptake into the human body. In addition, we have a pH response that's seen from the injection site where that injectate was introduced and then diffused away from thereafter. And then we have our turbidity being read throughout the duration of an assay to give you real-time updates on how much scattering is going on within that cartridge. And then those optical images are shown on your right. It's just a way to just demonstrate that throughout the duration of the assay, images can be taken on however small time scale the reaction or phenomenon are taking place. So hopefully that gave a brief update about the capabilities inside SCISSOR, what it's used for, and hopefully going forward, we're going to introduce to you a couple different case studies that go over some of these different application spaces and how it is used. So I believe I will be passing it off to Misha for our first case study about how Merck parameterized formulation for head-to-head comparison.
Yes, hello everyone. Thank you, Connor, for introduction. Yes, we'll provide some of our case studies that we've done at Merck with SCISSOR, specifically exploring the predictive potential or power and operational use of SCISSOR to streamline subcutaneous biologics drug development. And, you know, as the title states, we focus primarily on evaluating SCISSOR in the biologic space. However, as Connor mentioned, SCISSOR is meant not only for biologic, but for small molecules as well. But for the purposes of this presentation, we'll focus on biologics only. I think I have control now. So first, we're going to go over the case study number 1, which is ultimately looking at parameterization for formulation head-to-head comparison. So we wanted to know exactly what parameters to keep consistent or how to set up our experiments if we have multiple formulation candidates, and we would want to know which formulation performs better than the other. Other way. So for the objective overall, why we're looking at the subcutaneous biologics or subcutaneous mAbs in particular is that predicting bioperformance, specifically bioavailability of subcutaneous administered biologics, remains a large knowledge gap in drug development. The underlying causes of this clinical bioavailability spread among different commercial subcutaneous monoclonal antibodies, which is vary between 40 to 90%, are not entirely understood. Furthermore, there is no established reliable translation from preclinical species to human or a reliable in vitro method that predicts clinical performance. This lack of understanding creates uncertainty in formulation development, clinical dose estimation, and support an IV to subcutaneous formulation transition later in the development. So, as you all can see, there is an urgent need for development of biopharmaceutics bioperformance prediction models based on in vitro, in vivo, or in silico platforms. So here in this presentation, we discuss the predictive potential of power or operational use of one of the in vitro models of interest, which is SCISSOR, to streamline the subcutaneous biologics drug development. So for this case study 1, what we did, as the title states, we conducted an evaluation of different formulations of monoclonal antibody 1 in the SCISSOR N1 system, and the purpose was to determine if SCISSOR could distinguish between various formulations of this monoclonal antibody 1, while keeping the injection volume and formulation concentrations consistent. As a result, SCISSOR accurately and successfully differentiated among different monoclonal antibody 1 formulations as depicted in the figure 1A, and accurately depicted the fate of the formulation at the injection site. Data not shown here, but we will talk about it in upcoming slides. Assessment of the effects of the injection volume, which is figure 1B, and the concentration, which is figure 1C, on the release profiles revealed that only the volume of injection affected the release profile in SCISSOR. So, in other words, when we initiated this experiment, we first wanted to see if SCISSOR able to differentiate among different formulations of the same monoclonal antibody. And then we wanted to know what effect of volume or effective concentration of the formulation has on the release. And from that point, we saw that the volume of injection makes an impact on the release profile. So it gave us a clue that if we want to do the head-to-head comparison from one candidate to the other, we'll have to keep the volume of injection the same. So then for the case study number 2, in this case, we actually used another monoclonal antibody to do the head-to-head comparison and see how it corresponds to our analytical data and other experiments that we've done internally. So in this case, we conducted SCISSOR and 1 studies on monoclonal antibody 2, which underwent formulation changes ranging from minor, such as an excipient change, to major, which is a process change. And in the case of the process 1 versus process 2 formulations, SCISSOR yielded comparable release profiles with minor differences in the extent of the release at 24 hours, which is depicted in figure 2A, which was confirmed by a preclinical study. Additionally, in the formulation surfactant screening for monoclonal antibody 2, SCISSOR successfully determined the necessity of the surfactant in the formulation and demonstrated comparable release profiles between Paloxamir 188 and PS80 containing formulations in figure 2B, which actually further aligned with the results from analytical comparability. So from these 2 case studies, we were able to put this table together of summary of the formulation changes that can or cannot be assessed in SCISSOR. And as simple as this table is, we internally get a lot of questions from other groups regarding some of these things. So like I said, as simple as this table is, it's somewhat informative, especially for folks who are fairly new to SCISSOR and want to know how to use it and where to use it. So the changes depicted in green can be assessed in SCISSOR, and those changes are such as injection volume, change in the formulation composition, introduction of novel excipient, or change in the formulation attributes such as pH, osmolarity, viscosity, etc. However, at this point in time, things such as change in number of injections, change in the device, anatomical site of the injection assessment, or depth of the injection cannot currently be assessed in SCISSOR. So with that, I will pass this presentation to my colleague Aparna, who will go into, oh, actually, no, I'm so sorry. It's not Aparna.
Okay.
I'll go giving it back to Connor, who will present case study number 3.
Thank you, Misha. I think I can grab control. All right, so sorry for the slight delay there. So to sort of grow on what Misha just described, especially the additions of excipients and looking at different ingredients and formulations and how they affect the release profile of different biologics, we're going to be looking at that excipient effect using monoclonal antibodies. So the objective of this 3rd case study, if it will let me go forward. Yeah, there we go. Okay, so we have a denosumab injection, a commercially available formulation of denosumab, and we systematically removed or added excipients via dialysis. So dialyze overnight against specific surfactants, and then see the change in release profile that was exhibited thereafter within SCISSOR. The details on the dialysis can be seen in the table in the upper right-hand corner, but obviously we want to focus on the data that can be seen in the graph of the release curves. And so I think you guys can see my mouse. I can't see the PowerPoint little laser pointer, but I think you guys can see my mouse. So the light blue line is the stock base commercial formulation of denosumab. And the almost overlapping orange line is sort of our dialysis control, where we dialyzed against all excipients. So we didn't actually replace any excipients. It's just we wanted to test what the dialysis method was actually doing to the formulation. And it turns out it did change it slightly. It did increase the standard deviation of these injections slightly. However, we have, but we have almost overlapping release. So we were comfortable moving forward with this methodology. On the extreme side, we have the dark blue line, which is just the monoclonal antibody itself if all excipients were removed from the formulation. And you can see that much like when you don't add surfactants to mAb-based formulations, you get incomplete release, most likely due to aggregation or instability of the monoclonal antibody as it's introduced to a physiological space, or more dilute space than its formulation. When you have only the surfactant added to the formulation, we exhibited inside SCISSOR what we consider early release. And typically, that is reflux up the needle pathway due to surface tension or a viscosity issue or an injection issue, so to say, or observation that results in the pathway. So when we only have polysorbate 20 as the only excipient, it impacts almost the formulation to too much of a degree where that surface tension is most likely interrupted, and then it allows for reflux away from the injection site. So not delivered where you'd like. But the last observation I want to point out right here is that when we add back in the cryoprotectant agent, aka also a thickener, which would result in the orange line, you can see that the thickener actually removes sort of the cons of adding an excipient to a formulation. So even though it's not directly related to solving a surface tension problem, the addition of those cryoprotectant agents at higher concentrations that affect viscosity actually negate the sort of negative repercussions that come from the surfactant that we almost always have to add to a biologic formulation. In addition, we also took a blinded mAb. Some details can be seen on the right-hand corner about this blinded mAb, and then we systematically added different types of excipients to this formulation to see how the response would change. And so there's a lot of lines on this graph. I'm going to try to boil it down to only a couple that we have to really pay attention to. However, so we can see the dark blue mAb itself. And then much like with the previous experiments, when adding in a surfactant, we found specifically this time it was polysorbate 20 that still exhibited early release. You can see the large standard deviation on that light green right there. That was caused by the sporadic behavior of the injection. However, when adding polysorbate 80, we didn't find that early release was exhibited, and we had a nice, stable, reproducible release of our injectate. That followed closely to the pristine mAb solution that we had in the beginning. The other things I just want to mention is that when we were exploring with glycerol, which is typically used as a thickener, when it wasn't at high enough concentration to significantly affect the viscosity, because of its increased solubility of maybe the bulk formulation, we got very fast increase with very small additions of glycerol. So that was just an observation that I thought was interesting and that we intend on moving forward with and exploring how each of the solubility profiles of each of these cryoprotectants and how that can affect the release profile in different ways. And so I think with that, that's most of the major takeaways on this graph. I don't want to spend too long on this on this study, but if there's any questions, we can always come back and discuss all these excipients in detail. So, but moving forward, I do want to hand it off to Aparna, so she can talk about antibody precipitation within the SCISSOR platform and how they were able to characterize that.
Yeah, thanks, Connor. Good afternoon, everyone, and good morning. So for this next case study, we're going to focus mostly on the ECM cartridge and characterize the SCISSOR cartridge at the completion of a release study. in this particular study, what we did was we injected MAP1, which we discussed in the case study 1, into the cartridge and performed the SCISSOR workflow for 24 hours to monitor drug release. As you can see, there's usually a cloudiness or a precipitate that develops at the bottom of the cartridge when we inject a MAP. And this cloudiness is usually absent in a cartridge where we injected just the placebo, which lacks the protein active. So our immediate hypothesis was that this cloudiness that we see is indeed MAP related, which is what we wanted to characterize with this work. And I should mention that this cloudiness that we see is really MAP1 agnostic. We see similar findings with most drug injections. So to characterize this precipitate, we collected the ECM contents from the top, middle, and bottom fractions of the SCISSOR cartridge at the end of a 24 run, and we characterized the contents using different analytical tools listed here. We measure the concentration and turbidity of each fraction via absorbance at 280 and absorbance at 350 and 500 nanometers. We leverage size exclusion chromatography to monitor aggregation. We also leverage dynamic light scattering to understand the size distribution of the analytes in each fraction. And then lastly, we leverage a tool called analytical ultracertification to again detect any low molecular weight or high molecular weight species that could be present in each fraction. And then for all of these assays, what we ended up doing was collect the fractions diluted with the SCISSOR buffer to aid in the analysis. And as a control, we compared all our findings with the contents of a cartridge that was injected with just placebo. So for this first analysis, wherein we look at the concentration and turbidity, we pretty much wanted to get a confirmation on the mass balance. As you remember from case study 1, in the case of MAP1, we achieved about 70% drug release from the cartridge within 24 hours. And looking at the concentration of the leftover protein in the cartridge on the left graph over here, overall, we have about 34.5 mg of the protein that's left over in the cartridge. And this amounts to about 25% of the original MAP that was loaded in the cartridge. So that gives us a pretty good mass balance considering 70% of the drug was released. And when we look at the turbidity, as is expected based on the cartridge image that we showed in the previous slide, we see the highest turbidity at the bottom fraction of the cartridge compared to the top half of the cartridge. So with this particular set of assays, we confirmed that the top fraction of the cartridge has the least protein, whereas the middle and the bottom fraction have the most amount of protein content and also is more turbid. And in this next assay, we leveraged size exclusion chromatography to monitor aggregation in the cartridge. So again, over here, we took the contents of the cartridge, diluted it with the SCISSOR buffer, and injected it into the column. And each fraction or each type of a molecule is going to have different retention time within the column, which is attributed to the size of the molecule. So with the first graph on the left over here we're just doing a head-on-head comparison of the chromatograms of the contents of a cartridge that was injected with the MAP1 and the contents of a cartridge that was injected with placebo, which is the blue graph that you see over here. So pretty clearly we see that, the peak that we see is only observed in the cartridge that was injected with the active and not with the placebo. So this just confirms that any signals that we see is related to the MAP and could not be attributed to the ECM cartridge matrix or to the excipients that are present in the formulation. And then in the right graph, we're looking at the contents of the cartridge injected with the active in a more granular sense. So here we're looking at the top, middle, and bottom fractions of the cartridge. And essentially, we see a pretty clear difference in the amount of protein and aggregate formation. More protein aggregation or high molecular weight species are detected in the middle and the bottom fractions of the cartridge, which again, correlates well with the image that we saw in the first slide that I showed. And to pair with SEC, we also performed dynamic light scattering to monitor the size distribution of the analytes in each fraction. The way DLS works is that we shine basically a monochromatic laser onto our samples, and depending on the size of particles that are present in the solution, they scatter light differently. And based on the intensity of that pattern, you're able to estimate the size of different particles present in your fractions. So again, confirming what we saw in the SEC study, we detect the presence of larger size aggregates in the middle and the bottom fractions of the ECM cartridge, whereas the top half of the cartridge is usually has pretty small size particles present. And the last test we performed leveraged a tool called analytical ultra-certification that allows for the enhanced separation and detection of low molecular weight as well as high molecular weight species based on their sedimentation rate. And this sedimentation rate is related to the molecular weight of the species. So in this case, again, we take the contents of the cartridge diluted with the SCISSOR buffer, and then load it onto the ultracentrifugation column and spin it at 40,000 RPM. And then as the different molecules kind of sediment, we get the sediment based on the sedimentation coefficient, which is represented on the x-axis over here. And this is directly proportional to the molecular weight of the different molecules. So again, just another confirmation that we observe higher molecular weight species, which is indicative of protein aggregation in the middle and the bottom fractions of the cartridge, whereas we see lesser aggregates in the top portion of the cartridge. And then when we look at the just the peaks of the AUC, overall a wider peak seen with the bottom fraction is indicative of a more heterogeneous population. So we have more heterogeneous population present at the bottom of the cartridge compared to the very top. And in conclusion, all the ECM characterization studies, what they tell us is just a good mass balance of what is remaining in the cartridge is indeed the leftover protein that was unreleased. And over the course of the 24 hours, although the simulation is done at physiological temperature and pH, overall at the end of 24 hours, we do see some startings of protein aggregation, mostly in the bottom half of the cartridge. So with that, I can kind of hand it over back to Misha.
Thank you, Aparna. I'm going to request control. All right, so we're going to go over the case study number 5.
My mute button is missing.
Okay. So we're going to go over case study number 5, which is going to look into SCISSOR by predictive power. And in that case, we are looked and tested 2 commercial and 1 internal subcutaneous delivered mAbs with high, medium, and low clinical bioavailability values in SCISSOR N3 system. Okay, so yeah, this was the project that we performed last summer where we wanted to evaluate the biopredictive potential or power of the SCISSOR system. And ultimately, the project, what we did is that we took 2 commercial mAbs. One is a very high bioperformance. In this case, is a Praluent, which has bioavailability around 85%. One that is pretty much one of the worst ones, which is Kuntium. That is a European product. In the US, it's known as Silic. It has a low bioperformance and bioavailability of around 55%. And then we took one of the internal compounds, which had medium bioavailability, which was somewhere in between. And in this case, the objective was to determine if SCISSOR could accurately rank order these mAbs based on their clinical performance. We performed this experiments on SCISSOR N3 systems, thus we have actual standard deviations this time. And then we performed some statistical analysis for the drug release at up to 24 hours, as well as the AUC. And then we also looked at the drug release at 6 hours, as well as AUC. And the results shown here in figures 3A and 3B indicated that SCISSOR effectively and significantly ranked these 3 mAbs utilizing the release data up to 6 hours, percent drug release and AUC with statistical significance as predicted by their clinical performance. This was a very interesting and is a very good study to do because it showed that it has this potential to differentiate among different performing mAbs as based on their clinical performance. The thing that we derived from the study was the fact that, as you guys can see, if we look at the release profile and the AUC up to 24 hours, we do not see any significant differences among these mAbs. And if some of you are familiar with SCISSOR and its development work, one of the publications actually came out a few years back that looked into the hyaluronic acid degradation within the ECM. And that's why the initial studies that were performed in SCISSOR were looking at the drug release pretty much up to 6 hours. The reason why we looked at 24 hours is because from Pion, the recommendation is that the max amount of time with the standard ECM is 24 hours. That's why we use it, the 24 hours for all our experiments. However, in this case, we wanted to, since we're looking at the biopredictive potential of this instrument, we looked at 24 hours as well as at 6 hours. And at 6 hours is where the hyaluronic acid is still stable. We actually saw some statistical significance and saw that SCISSOR is able to differentiate or rank order these mAbs according to its clinical performance. That would be actually a good segue to the next section by Connor, who is going to talk about different ECMs that Pion is currently working on to potentially increase this resolution and potential to distinguish among mAbs that are not only on the extremes, like in this case, best, worst, and somewhere in the middle, but could potentially distinguish mAbs that are closer to each other on their clinical performance. So for the conclusion for the Merck side of the experiments, we derived that SCISSOR system is able to capture the behavior of the formulation at the injection site. So it detects any precipitation, aggregation of formulation at the simulated site of the injection via transmission and imaging data. It has a capacity to differentiate formulations with different release profiles, which is valuable for excipient of formulation screening and formulation development, and has a potential to obtain this IV-IVR, or in vitro in vivo relationship, based on the release profiles in the preclinical and clinical assessment. However, the translatability of SCISSOR results and its predictive power to the clinic has not been fully demonstrated, as supported by multiple publications in the literature, in the reference slide that I will provide next, and our internal analysis. Thus, the predictive power of SCISSOR needs to be investigated further with more examples of different types of subcutaneous biologic products and their corresponding clinical data. To establish this IV-IVR, SCISSOR could be routinely incorporated in the subcutaneous biologics drug development workflows.
All right.
So in this case, the acknowledgement and references from the Merck side. So I would like to thank direct contributors that are depicted here: Mikawai, Susan, Yash, Jing, and other folks who were involved. And then for the references here, we actually provide like all of the relevant SCISSOR relevant publications, as well as some of the instruction material and some of the material that we use from Pion towards our experiments, as well as FDA labels for the commercial mAbs that we used in our experiments. And with that, I will pass back to Connor, who is going to talk a bit more about different types of ECM that would elaborate on our case study on the biopredictive power of SCISSOR.
Thank you very much, Misha. Not just for the little intro, but also for showing all the incredible work that Merck's done up until this point, both Misha and Aparna. But now, as Misha and Aparna pointed out some strengths and shortcomings within, or not shortcomings, but the applicability of SCISSOR within different types of workflows. Here at Pion, our goal is to develop the instrument to a point in which it can be more sensitive for separation or rank order of formulation or rank order of excipient screening. Really briefly before we end, I'm just going to go over quick examples of what the other ECMs and what our development pipeline sort of is looking like right now. This is an image from a recent publication we had this year in the European Journal of Pharmaceutics. These are injections of dextran at 40 and 70 kilodaltons for the release curves. And then the 70 kilodalton boluses in diffusion can be seen in the images on the right-hand side. So the three ECMs that we have are on the top. We have a hyaluronic acid-based ECM. We have a hyaluronic acid-based ECM again, but with the inclusion of collagen expressed from human fibroblasts. And then on the bottom, we have a sort of an extended release format of that HA hydrogel. So the extended release gel is designed where you have enough room to inject into and form sort of a bolus or an injection site. And then what happens is that bolus is actually kept in situ for the entire duration of the experiment. And this experiment has been ran up to 1 week. And this has been exhibited up until that time. Really quickly, just to talk through the images, in addition to SCISSOR showing that we have a molecular weight relationship between release curves, which of course is important characteristics, but not really too much of a fun scientific discussion. But the bolus formation in the strictly glycosaminoglycan-based ECM, you can see bolus formation within that gel, and then homogeneous diffusion on a front, most likely based on density from the top of the cartridge to the bottom of the cartridge, almost like a gravimetric chromatographic method. With the inclusion of collagen from human fibroblasts, so it's going to be a slight mixture. You can see that diffusion pathway changes slightly. So the inclusion of all those partially positive molecules and longer, more surface area to interact with, that injectate spreads out in a more homogeneous way throughout the entire cartridge, at least in the upper half where that bolus is located. And then that diffusion seems to stay on that natural pathway throughout the duration of the assay. Not only showing that collagen absolutely is playing a role in changing the maybe architecture or mechanical characteristics, which we also saw in the publication, but it also affects the pathways in which these injectates actually diffuse out, most likely based on the electrostatics and the slight change in pore size. Oops. And then lastly, with that extended release model, you can see that bolus is conserved. However, it's not really a natural bolus shape at this point, but if you were able to monitor, if you were wanting to look at the interaction between an injectate and that matrix over a long period of time to ensure that you do catch all the post-injection phenomena that could happen in an injection site, that is designed for that. The XR is designed for that application, excuse me. And just really quickly, to more highlight the inclusion of monoclonal antibody-based injections within this, within these platforms, you can see here sort of a view on the aggregation behavior that can be seen within the collagen-containing matrix after the injection of a commercially available formulation of denosumab. The release curves within the collagen, because the collagen is actually going to stabilize the overall architecture of the glycosaminoglycan-based gel. You do not get either diffusion of as much bulk HA into the receiving chamber, so there's less spectroscopic apparition that can be seen as compared to the strictly glycosaminoglycan-based ECM. But different collagen concentrations actually resulted in the same total overall release, although different kinetics along the way. And these types of processes are what we're looking into right now to ensure that we know how each of these processes is actually affected and what it means in terms of a clinical response and a clinical relationship. So with that, I would just like to acknowledge our seminar partners, our webinar partners at Merck with Misha and Aparna. It's been a pleasure working with them, and the work they showed is extremely elegant and straightforward. And we'd also like to thank Bath University, Randy Mersney and Kate Gridley there. They've been involved in the SCISSOR development thus far, and have to give them their due credit. And just there are some few validation studies we've done with either with Bath University involved or others involved, as well as in-house. You can see that last publication here. That's sort of, if you are interested, those are 3 good publications to dip your toes inside the entire SCISSOR arena. So with that, I want to thank everyone for their attention, and we are welcome to take any questions you have. Oops. I'm not sure why that happened, but anyways, I'm trying to forfeit control and get to Q&A.
Sorry, Connor,
Yeah.
I was trying to take controls back.
I forfeited them, don't worry.
So let's take the first one here. What is the cutoff of the membrane?
Yeah, sure. Sorry. We're in the same room, so we're on each other's audio. The cutoff of the membrane is 5 micron pore size. There's no official molecular weight cutoff for the membrane. It's just a large pore size of 5 micron.
Okay.
May I add something here?
Sure,
Yeah, so we get this question a lot as well, and Amy, Connor, please correct me if I'm wrong, but a lot of folks think that the membranes, that's what's govern the diffusion of your formulation to the sink buffer, where in reality, your membrane size or pore size or membrane itself should not have any interaction with your formulation. In fact, that membrane acts more of a physical barrier to maintain or keep the ECM within the cartridge. All of your interactions should be happening between your formulation and ECM itself. And once those interactions happen, the protein or the formulation is freely diffusing out into the sink conditions. And in fact, from what I remember in the original instruction manuals, if your protein does have any interactions with the membrane, you're supposed to change the membrane to a different one.
Thank you both. Next question. For the dextran study for ECM analogs, did you measure the release profile of dextran formulation? And what was the formulation vehicle for dextran?
They were just fluorescently tagged dextran molecules of different molecular weights. They were not full formulations. So the only difference between, they were buffered solutions, and the only difference between them was the molecular weight in which dextran was included in that injectate. But yeah, we did run those experiments, not with formulations, but in short, there's really not much to go over, but you have a molecular weight related release curve, which would happen with most injectates, where you do have the same API at varying molecular weights, and we were able to show that quite significantly within SCISSOR. So that's dextran.
Thank you, Connor. Question for Misha. Thanks for presenting the data, great observations. My question is, did you observe any change in release when injecting mAb closer to the membrane as compared to the center of the cartridge?
That's a great question. Actually, I don't see this question from my end, but whoever asked this question probably knows me personally, because I talk about this a lot. So this is a very good question because, as I mentioned earlier, the formulation that you inject has to interact with ECM. And that means that you need to inject that formulation directly in the middle of your cartridge, so your ECM is able to encapsulate the formulation for the proper interactions. If you do have injection that will place the injection somewhere to the side of the cartridge, we did notice difference or variability in release profile, and sometimes even the instant release, because you miss these interactions within the ECM. And with the SCISSOR N1 and even N3 systems, you still use commercial needles, right? And one thing that I want to point out there is that pay attention to your needle, because sometimes that needle is not straight. Sometimes it's slightly curved to the left, to the right, down, or up. So if possible, select the most straight needle as you can, because that way even though you inject the formulation according to the instructions, that will ensure that the placement of the injection will be to the middle of the cartridge as possible, or in the middle of the cartridge as possible, so ECM is able to engulf that formulation.
Thank you. Next
I can just add to
question.
that for like
Oh, sorry about
1
that.
minute.
Go ahead.
In case anyone's wondering about like all those different parameters and how they would affect diffusion, there's a great paper I think out at the University of Kansas that does some Monte Carlo simulations of the SCISSOR cartridge, and so I think that actually shows a great. It's just a more, if people want to look into it, there is that paper out there, and it does show that yes, it does change, but the small deviations in the middle probably are insignificant compared to if you are injecting it significantly more towards that membrane. So that's just my anecdote.
Hey, thank you, Connor. Why does the pH increase with time?
I assume this person is talking about the pH monitoring at the injection site with that graph we showed where the pH drops down and then back up. That's because that injectate had an acidic bias. It was an acidic formulation. So when you inject, you get to see that pH drop down to a lower pH. But then over time, the bicarbonate buffer, which is pH controlled by the carbon dioxide at 7.4, can passively diffuse through that membrane and through that hydrogel the entire time. And so as the injectate diffuses away and that carbonate buffer diffuses back within the ECM, you just get to watch that pH response.
Okay, next question. How were the 3 layers in the cartridge separated successfully for analysis of protein aggregation without being merged together?
Yeah, essentially, the way that the 3 samples were collected is the top 1.5 ml was considered the top fraction, the next 1.5 ml aliquot collected was the bottom fraction, and the last 2 ml that we collect is regarded as the bottom fraction. And if you have any experience working with a SCISSOR cartridge, essentially when you remove it from the instrument, as long as you don't agitate it enough, the layers remain pretty intact.
Yeah, the ECM itself is very viscous in the beginning, and even in the end of the experiment, it still remains this gel-like structure. So it's somewhat easier to separate fraction by fraction.
Okay, I think this one may be for Alparna again. Maybe I missed it, but are you doing DLS in situ or drawing samples from the injection chamber? Any impact on DLS measurements from the ECM?
Yeah, in this case, again, we're doing the measurements once we remove the fractions from the chamber, so we're not doing in situ DLS measurements. For the DLS run in particular, we did not have the placebo control, so I cannot fully comment on whether any aggregates that we see in the DLS, if they are attributed to the ECM or they're solely protein related. But again, overall, with each fraction that we collect, the ECM is the common matrix in each fraction that we collect. So the increasing trend of the aggregates that you see could be more related to the protein and not the ECM, which is kind of at a standard baseline in each fraction.
Okay, next question. Have you tried using PBPK modeling to understand the relationship between the in vitro profiles and those observed in clinical studies?
I guess I can comment on that or take a stab at it. Good question. Currently, there is not that many PBPK modeling platforms that evaluate biologics or subcutaneous biologics. We did look into GastroPlus biologics module, and in fact, GastroPlus biologics module actually has SCISSOR as input. However, this biologics module is in the very beginning stages or rudimentary stages, so it does not have the same capabilities as it would have, you know, in oral space and whatnot, because we in biopharm use GastroPlus a lot. So for those reasons, we did play with it a little bit. However, there is still work to be done to improve that model that we currently, you know, help Simulation Plus folks to work with it through this, as well as some other companies, and I'm sure academia as well. Other than that, I don't think that there are many biologics, subcutaneous biologics in silico platforms. And actually, to my point that I made during presentation, there's an urgent need for those predictive tools, you know, in silico, in vivo, or in vitro. So folks trying to improve them, folks trying to work on them, but currently we just don't have established capabilities already in that space.
Okay, thank you all. We've run out of time. We still have many questions in the queue. We apologize. We won't be able to get to them during the live session, but we will answer them offline. I want to thank everyone for joining us today. And I also want to thank all of our presenters in putting all the information together and presenting all these great case studies with us today. Again, the webinar is being recorded, so it will be available online within 24 hours or so. And at this point, I'm going to end the webinar session. Thank you all for your participation and have a wonderful rest of your day.
Thanks,
Goodbye, all.
everyone.
Thank you, everyone.
Thank you, everyone.
Bye now.